एयरपॉड वाले भी
तुमसे
एप्पल एयरपॉड वाले सॉरी एप्पल वाले भी कनेक्ट हो जाएंगे नॉर्मल वाले जो इसमें लगता है चार्जर वाले
चार्जर वाला
इसका जो चार्जर वाली पिन होती है तुम्हारा वैसा वाला चार्जर अरे इसका भी चार्जर तुम्हारा कैसा है सी टाइप है क्या
इसमें सही है इसमें भी तो होना चाहिए इसमें मैग्नेटिक चार्जर होता है ये मैग्नेटिक चार्जर वाला पट यार 3 बज गए Good afternoon everyone and welcome to today's session. I guess we'll start in 5 minutes so that more people can join in. Okay.
पटी गई है जल्दी बताओ
इसका स्विच ऑफ कर दो वहाँ तो टीवी अपने आप ही चलने लगेगा
हाँ क्योंकि वो पूरा ऑफ हो रहा है ना
पता नहीं क्या
पूरा ऑफ होकर के पूरा ऑन हो रहा है तुम्हारा हम ये कनेक्ट कर रहे हैं अपने से
वो आया है सुन लो वो आया है इन्वर्टर वाला वो ठीक कर रहा है वो बार बार कनेक्शन जाएगा इसीलिए जा रहा था इसीलिए मैं सोची थी कि अपने से कनेक्ट कर दूँ जैसे ही वो होगा तो वो कनेक्ट होगा ये होगा तो ये कनेक्ट हो जाएगा नहीं वो अपने से कन्वर्ट होगा फिर आएगा इन्वर्टर वाले तो वो भी आना पड़ेगा
Good afternoon, everyone. I hope my slides are visible to you guys, and welcome to today's session. So, in the last class, in the last few sessions actually, we were discussing a lot about lipids itself, right? So, we ended our discussion on sterols. So, we studied about different types of sterols which are present in living system. Bacteria doesn't have any sterol compounds in their membrane, but other microorganisms do have, but not exactly cholesterol. We have cholesterol. Fungi have similar molecule called ergosterol, and then plants have different kind of sterol called beta cetosterol, right? So all these compounds are structurally very similar, as you can see, and we have studied about their roles also, like what role do they play in being present in the membrane apart from other kind of lipids. The major lipid were phospholipids. After that we studied about sphingolipids and all, right? So that marked our discussion for lipids. So few pointers before concluding our lipid discussion with respect to membrane. It is for example just little bit of like summarizing lipids for with respect to biological membrane. So unlike protein and nucleic acid compositions that are defined, lipid composition can vary considerably with different organisms, different tissue in the same organism, and different organelles within a cell, different membrane domains within a cell membrane, and even different phospholipid molecular species in the same cell. So you know that this pointer is pretty evident, right? If you remember our basic membrane distribution, like lipid distribution in the membrane, you can clearly see that even within a cell there is huge diversity of lipids, right? So if within a cell there is a huge diversity in lipids, then between the cell, between the organisms, the variability will be huge. So this is one of the major reasons that we are not able to clearly classify all the lipids which are present. Okay. Though we have some idea how to classify them, but yet some lipids will always fall apart in those classes, and moreover we have classified only the major lipids, right? We haven't even classified small lipids which play significant role. So it's an active area of research. Then another basic statement about lipid distribution in membrane is lipid composition can vary with diet, environmental conditions, and age of the organism. Okay, the major structural lipids are phosphatidylcholine and then phosphatidylethanolamine. There are no cationic lipids. My question will be to you guys, why there are no cationic lipids? Try to search it up, and if you guys are not able to find the plausible answer, do write it to me. I will explain it to you. Okay. The anionic lipids, that is phosphatidic acid, phosphatidylserine, phosphatidyl'inositol, phosphatidylglycerol, and cardiolipins, are all found at lower level than the structural lipids, that is phosphatidylcholine and phosphatidylethanolamine. Phosphatidylcholine is a Zwitter ion, true Zwitter ion, and has no net charge, while PE, that is phosphatidylethanolamine, is almost a full Zwitter ion that has a slight net negative charge. Cholesterols can vary from trace amounts, inner mitochondrial membrane, to being the major polar lipid, over 50% mole percentage in the plasma membrane, depending upon the particular membrane. Cardiolipin is found almost exclusively in the mitochondrial inner membrane. Free unesterified fatty acids and lyophospholipids are found in very low amounts where they adversely affect membrane structure. High enough amounts of free fatty acid or lyophospholipids will kill a cell through the detergent effect. Fatty acids are distributed unevenly between the two phospholipid acyl chains. The SN1 chain are predominantly but not exclusively saturated, while the SN2 chains are mostly unsaturated. Two chains are unsaturated, 18, 20 or 22 carbon. This is not a hard and fast rule, but this is generally observed. Okay. The large number of unsaturated acyl chains means that on an average at physiological temperature, most if not all of the membrane will be in melted or fluid state. This makes complete sense, right? Why? Because the membranes are explained via fluid mosaic model. So the fluid part of the fluid mosaic model actually comes from this, that most of the lipids which are making the major part of the membrane are saturated at room temperature. So if they are saturated, they are unsaturated, indicating that they will be fluidic in nature. Correct? So overall, this marks the end of our discussion with respect to lipid composition of the membrane. Now we'll start with the proteins, which are another major component of the biological membrane. So all of you know what proteins are, right? Proteins are a polymer of monomers called amino acids. So what exactly amino acids are? Proteins are polymers of amino acids, with each amino acid residue joined to its neighbor by a specific type of covalent bond. And this specific type of covalent bond is called a peptide bond. Okay. Proteins can be broken down or hydrolyzed to their constituent amino acids by a variety of methods, and the earliest studies of proteins naturally focused on the free amino acid derived from them. There are 20 different amino acids which are commonly found in proteins in living system basically, but they are not limited to the number 20. There are other amino acids also present, but they are not predominantly forming proteins in the living system. They might be present in some special places, playing some important function. So if we're gonna dig more into amino acids, why? Because at the end of the day, if we want to understand proteins, which are another component of a membrane, that component can be easily understood if we know the monomeric side of the polypeptide and protein. So what amino acids are? You guys might be aware of it, but let me freshen up your memory. That amino acids share some common structural features, right? And what are these common structural features? All 20 of the common amino acids are alpha amino acids. Okay, they have a carboxylic group and an amino group bonded to the same carbon atom, the alpha carbon. They differ from each other in their side chains or R groups. The side chains are called R groups here, R being variable in all 20 amino acids, which vary in structure, size, electric charge, and which influence the solubility of the amino acids in water. So if I show you the general structure of amino acid, you guys might remember this, that a C alpha will be in the center. On one side, amine group, that is NH3, will be attached. Another group, carboxylic acid, will be attached to the C alpha. Then the R, the variable side chain, will be attached here. And last but not the least, an H is present here for the completing the valency. So this is the general structure of a protein. Excuse me. So this is the general structure of a protein. We are going to dig more into it. So all of you remember your 20 amino acids? How many of you are can name all the 20 amino acids? Probably some of you can do this, but let me freshen up your memory again a little bit with some clear classification of amino acids. Okay, so amino acids can be classified by R group. Okay, and by R group I mean the variable chain which we are showing. So if we try to classify the amino group on the basis of side chain, right? So you can classify amino acids based upon any criteria. For example, weight, net charge, or say whether they are aromatic, non-aromatic, right? So overall the molecule. So if we look the classification which is based upon the R group or the side chain of the amino acid, we can somehow broadly categorize and classify the amino acids as non-polar aliphatic, aromatic, polar but uncharged, positively charged amino acids, and negatively charged amino acids. So in these broad 4 categories we can categorize our amino acids, our 20 amino acids. Okay, here we are only talking about the classical 20 amino acids. So in the non-polar aliphatic R groups we have 7 amino acids. Glycine, in which the side chain is, see, you can look at the backbone of the amino acid. It is NCC, right? So here N side will be the amine side, the C is the carboxylic side, C alpha is there, and another H is already associated. So this structure, this whole top structure, wait, let me have a pen. Yeah, so this whole structure is same in every amino acid, not only in aliphatic but all 20 amino acid, right? The only variability we are getting is from the side chain, this, the highlighted portion, if you can see. So this is the variability which we are getting in an amino acid, and these side chain or the R group, it defines the property of an amino acid. Okay. Oh, I'm sorry. Yeah. I hope you guys can hear me. There was some technical issues. Yeah, I'm sorry for the technical issue. Yeah. Oh God. I hope you guys can see my screen. Okay, so yeah, so what I was saying, I'm saying that if you have 20 amino acids, this backbone will remain same and only the R group will vary, and this R group is the one which is providing the basic characteristics to these amino acids and which perform different function depending upon the surrounding conditions, right? So if we talk about non-polar and aliphatic R groups or amino acids which are non-polar and aliphatic, we can see the simplest amino acid will be glycine. Why? Because the R group is basically not a functional group, not a big compound or molecule attached to the R position. It is simply an H. So this is the smallest amino acid which you can have. The next amino acid which is non-polar and aliphatic is alanine. So as you can see, alanine have a CH3 group in place of H or say R, which we are talking here, right? So this will be another non-polar aliphatic amino acid. Next will be proline, and proline is a special amino acid. As you can see that it makes a bond with the amine side of the backbone itself. There are no amino acids, at least these 20 amino acids which we are focusing on, that any amino acid in which the amine side or the N side of the peptide or of a residue is not free. Only in proline it is bound, and in every other amino acid the amine side or the N side is not free. Next one in the non-polar and aliphatic will be valine, and as you can see, this is the functional group which is present in the valine. Next will be leucine. You can see that it is again a non-polar and aliphatic amino acid with CH2, CH, CH3 and CH3 in place of R. The next will be isoleucine. Isoleucine is also similar to leucine, is just the arrangement of methyl group is different in comparison to leucine. Then the methionine. So here methionine, you can see this is the first amino acid which you guys are encountering which has a sulfur apart from the basic CH and NO molecules. So methionine is a sulfur containing amino acid. So when I am talking about classification of amino acids, so this particular classification is for when we are only considering the side chain. Imagine if you want to classify amino acids as any other way. So you can say sulfur containing and sulfur non containing, right? You can say having a ring structure, not having a ring structure. So there are multiple ways to classify these compounds. So the classification on which we are studying the classification of amino acid is based only upon the side, the variable group or the R group. Okay. Now if you look at the aromatic amino acids, the amino acids where the R group is aromatic in nature. So we have 3 such amino acids which are aromatic in nature. First one is phenylalanine, and you can see the structure of phenylalanine here. The other one is tyrosine. You can see that tyrosine has this ring structure associated in place of R group. Then tryptophan, and you can clearly see that tryptophan is kind of bulky amino acid with having a extended aromatic ring associated with in place of R group. Next category will be polar but uncharged amino acids, and here when I am saying amino acid, I mean only the R group of the amino acid. So polar but uncharged, there are 5 such amino acids, that is serine, threonine, cysteine, asparagine and glutamine. So all these 5 amino acids, they are polar in nature, but they do not have any charge associated with the side chain at physiological pH. So describing the property of a amino acid or amino acid residue, whether they are charged, they are positively charged, negatively charged, polar, non-polar or uncharged, it has to be with respect to some conditions, right? Some pH, because the moment we change the pH, they all are functional groups. So depending upon the surrounding, these amino acid residue, either they will gain a proton or they will donate a proton, right? So whatever charges we are showing here, exactly the structure which you guys can see on your screen is present only at the physiological pH. The moment you change the pH, the property of these amino acids or say the side chain R group varies. So if you look at the serine amino acid, you can clearly see that this is the first amino acid which you guys are encountering which has non-aromatic OH associated with it, okay, which means it can make hydrogen bonds. The next one will be threonine, and you can see the structure that it also has OH. So wherever the OH is present, we can somehow speculate and say, and we have also observed in the literature and in practicals also, in experiments also, that these OH are the ones which are responsible for modifications of a protein, okay. Then cysteine. Now cysteine is another amino acid which you guys can see which has sulfur associated with it, okay. So these sulfur containing amino acids, they play a critical role, and they are present in some special positions, generally special positions in the in protein. So cysteine is the one which can make disulfide bond. So don't worry about the details which I am saying, and you might think that you guys are not able to follow. I have mentioned all these details in the coming slides, and we're gonna go in every category and we're gonna see some of their special properties and the basic biochemical nature these categories provide to a protein. Then next amino acid which is polar and uncharged is asparagine, and you can see the R group which is depicted here. The next polar and uncharged R group is present in amino acid glutamine, and you can see the structure, and you can also relate how these amino acids are related to each other. So if you look at leucine and isoleucine, as I've told you, the only difference between leucine and isoleucine is the placement of the methyl group. Similarly, if you look at the asparagine and glutamine, you can see that their R group is somewhat similar, except that there is presence of one CH2 group. That's it. So if you want to relate and learn the structure of amino acids, you can learn them on the basis of their reference with the similar amino acid. This will help you in learning their structure. The other category of amino acids based upon the side chain or the R group is positively charged amino acids or positively charged R group. So it has three such amino acids which are positively charged: lysine, arginine and histidine. So lysine, as you can see, it has extended R group, right? And then arginine also has extended R chain. Then last but not the least, in the positively charged R group amino acid, there is histidine, and you can see histidine is also a special amino acid, and we're gonna see why I'm calling it a special amino acid in a while. Last category, based upon the properties of the R group or the side chain, they are negatively charged amino acids or are negatively charged R groups. They are present in aspartate, and you can see the functional group is CH2COO negative, and glutamate, that is CH2CH2COO negative. If you want to make some relation with their structure, you can relate the structure of aspartate and glutamate to their counterpart polar and uncharged R groups, that is in asparagine and glutamine, and you can easily learn their structure based upon, if you learn the structure of among these four, if you can learn the structure of any two amino acids, and then you can derive other amino acids structure. Now we'll see some properties of these amino acids at the end of the day. So the first category which we were looking at was the non-polar aliphatic amino acids, right, or R groups. So what were the amino acids which were present? There were 7 amino acids which were present in this category. Those 7 amino acids are glycine, alanine, proline, leucine, isoleucine, valine and methionine. Okay. So non-polar aliphatic R groups, the R group in this class of amino acids are non-polar and hydrophobic in nature. The side chain of alanine, valine, leucine and isoleucine tend to cluster together within a proteins, stabilizing protein structure through the hydrophobic effect. So the amino acids what we are looking at is alanine, valine, leucine and isoleucine. So alanine, valine, leucine and isoleucine. So these 4 are the amino acids which are non-polar and hydrophobic in nature. So these amino acids, what they'll do is they will club together if they are present in a peptide or in a protein via hydrophobic effects. Okay, and then they provide structural stability to the protein. Glycine, which I have told you, has the simplest structure, although it is most easily grouped with the non-polar amino acids. Its very similar side chain makes no real contribution to interaction driven by the hydrophobic effects. Methionine, one of the 2 sulfur containing amino acids, has a slightly non-polar thioether group in its side chain. Okay. Proline has an aliphatic side chain with a distinctive cyclic structure. The secondary amino acid imino group of the proline residue is held in a rigid conformation that reduces the structural flexibility of polypeptide region of the containing proline. So you remember I specifically mentioned about the proline, that it is this amino side of a amino acid is not free, unlike other amino acids. So wherever the proline will be present in protein, it will provide the rigidity to the structure. So if you say, if it is providing rigidity, indicating that the protein will be less flexible in such places, correct? So wherever the protein needs to bend, or there is a requirement of a peptide chain to bend or change direction or have a small kink associated in the protein structure, most of the times a proline molecule or a proline residue will be present there. So let me help you guys in remembering the amino acids by their properties which is imparted by the R group. So now we have discussed about the non-polar aliphatic R groups, right? So what are the amino acids in non-polar aliphatic R groups? First will be glycine, leucine, valine, isoleucine, methionine, proline, alanine. Okay. Coming to the next category was aromatic R groups, right? So aromatic R groups have phenylalanine. They had 3 amino acids. Remember this chart. So we have 3 amino acids in aromatic R group category. So aromatic R groups: phenylalanine, tyrosine, and tryptophan, with their aromatic side chains are relatively non-polar, right? Meaning hydrophobic. All can contribute to the hydrophobic effect. The hydroxyl group of tyrosine can form hydrogen bond, and it is an important functional group in some enzymes. So what we are talking about? We are talking about this tyrosine. Near the laser pointer. Yeah. So we are talking about tyrosine. So tyrosine has a OH group associated with the benzene ring, right? So this OH is a functional group and it can make hydrogen bonds, and hence it is present in places where there is some like proteins. Enzymes are also protein, right? So enzymes, wherever there is some functional role, it is observed that that tyrosine molecule will be present. Tyrosine and tryptophan are significantly more polar than phenylalanine because of the tyrosine hydroxyl group and the nitrogen of the tryptophan indole ring. You might not understand this right now, but if you know the structure, you can see that what that statement means. The statement means that tyrosine and tryptophan, they are more polar, right? This is what it says. This is more polar in comparison to phenylalanine because there are functional groups like OH and amine. NH group is associated with these residues, whereas there is no such functional group associated with the phenylalanine molecule or residue. Then tryptophan and tyrosine, and to a much lesser extent phenylalanine, absorb ultraviolet light. This accounts for the characteristic strong absorbance of light by most proteins at a wavelength of 280 nanometers, a property exploited by researchers in the characterization of protein. So what it means? So here you are looking at the absorbance curve by different aromatic amino acids. So here you can clearly see that at an around 280 nanometers, both tyrosine and tryptophan, both of them shows high absorbance, while though phenylalanine is also an aromatic amino acid, but it shows very less absorbance near to 280 nanometers. So this property can easily be exploited for various characterization assays of a protein. Okay, so I will try that you guys can remember all the 20 amino acids by the end of this class. So till now we have discussed 2 categories which are classified based upon the R group. The first category was non-polar and aliphatic amino acids, and the other was aromatic amino acids. In non-polar aliphatic amino acids, there are 7 amino acids. First one being glycine, alanine, valine, isoleucine, methionine, proline, and leucine. Okay, the other one was aromatic. It had 3 amino acids starting from phenylalanine, tyrosine and tryptophan. The next category is the polar but uncharged amino acids, or the side chains of amino acids which are polar and uncharged. So polar uncharged R groups, the R groups of these amino acids are more soluble in water or more hydrophilic, okay, than those of the non-polar amino acids, because they contain functional groups that form hydrogen bond with water. This class of amino acid includes serine, threonine, cysteine, asparagine and glutamine. The polarity of serine and threonine is contributed by their hydroxyl groups, and the polarity of asparagine and glutamine is contributed by their amide group. What it means. So we are talking about this category. So the first category was non-polar aliphatic. I'm going to repeat all the amino acids which were present in this category. There are 7 amino acids which are non-polar and aliphatic in nature. They are glycine, leucine, isoleucine, valine, methionine, alanine and proline. The next category was aromatic amino acids. They were phenylalanine, tryptophan and tyrosine. The category which we are talking about right now is the polar and uncharged amino acids. It has 5 such amino acids, and these are serine, threonine, cysteine, asparagine and glutamine. Among these, the polarity or the polar nature which is provided by the R chain, the group which is associated with it is the OH group in the serine threonine, while asparagine or NH2 group is present in asparagine and glutamine, which provides the polarity in the polar but uncharged amino acids. Cysteine is an outlier here because its polarity contributed by its sulfur hydryl group is quite modest. Cysteine is a weak acid and can make weak hydrogen bonds with oxygen or nitrogen. Okay, but yet cysteine is one of the important amino acid because this is the only amino acid which is able to form disulfide bonds, and we're going to discuss this in the later slides. So asparagine and glutamine are the amides of 2 other amino acids also found in protein, aspartate and glutamate respectively, to which asparagine and glutamine are easily hydrolyzed by acids or base. Okay, cysteine is readily oxidized to form a covalently linked dimeric amino acid called cysteine, in which 2-cysteine, look at the spelling, right, cysteine is a residue. Okay, when it is covalently bound with another cysteine residue, that is cysteine residue, this dimeric form is called cysteine, that is cysteine, cysteine, in which 2 cysteine molecules or residue are joined by a disulfide bond. The disulfide linked residues are strongly hydrophobic, that is non-polar. Disulfide bonds play a special role in the structure of many proteins by forming covalent links between parts of a polypeptide molecule or between 2 different polypeptide chains. So the amino acid which we have studied till now in the 3 categories, the first category was non-polar aliphatic amino acids, next was aromatic amino acids, and right now what we have discussed is polar and uncharged amino acids. So in non-polar aliphatic amino acids, we had 7 amino acids, namely glycine, alanine, proline, methionine, leucine, isoleucine and valine. Okay, in aromatic amino acid category, we had 3 amino acids, that is phenylalanine, tyrosine and tryptophan. Current category which we have discussed, which is polar but uncharged, we have 5 such amino acids. It will be serine, threonine, cysteine, asparagine and glutamine. Okay, so we are going to discuss next 2 categories which are left. they are positively charged or basic amino acids, the R groups, and the negatively charged acidic or the side chains are acidic in nature. So positively charged or basic amino acid, the most hydrophilic R groups are those that are either positively charged or negatively charged. The amino acids in which the R group have significant positive charge at pH 7 are lysine, which has a second primary amino group at the position on its aliphatic chain. Arginine, which has a positively charged guanidinium group, and histidine, which has an aromatic imidazole group, as only common amino acids having an ionizable side chain with near neutrality. Histidine may be positively charged, that is protonated, or uncharged at 7. Histidine residues facilitate many enzyme catalyzed reactions by serving as proton donor acceptors. Negatively charged amino acids, the two amino acids having R groups with a net negative charge at pH 7 are aspartate and glutamate, each of which have a second carboxylic group. So you can see the positively charged amino acids, we have 3 such amino acids which are positively charged, that is lysine, arginine and histidine. Among these 3, lysine is the one which is highly positive. Okay, and in the negatively charged amino group we have aspartate and glutamate. So let me try to help you out in remembering all these 20 amino acids with their R group property. So first we discussed about non-polar aliphatic amino acids. So in non-polar aliphatic amino acids we had 7 amino acids, and what were those 7 amino acids? Those were glycine, valine, alanine, proline, leucine, isoleucine and methionine. Next category was aromatic amino acids. In aromatic amino acids we had 3 aromatic amino acids. They were phenylalanine, tyrosine and tryptophan. Next category was polar but uncharged molecules, right? Uncharged residues. We had 5 amino acids in this category. They were serine, threonine, cysteine, asparagine and glutamine. Next category was charged amino acids, right? So positively charged amino acids and negatively charged amino acids. In positively charged amino acids we have 3 amino acids, that is leucine, arginine and histidine. In negatively charged amino acids or side chain which are negatively charged or acidic in nature, they are aspartate and glutamate. So I hope that you guys can remember these properties which are associated with amino acid residue, right? Let me show you guys some basic video which will summarize all the learnings which we have learned in today's class. So let me know if you guys can see my screen and hear the audio also. I hope you guys can see this.
Proteins are vital for the normal function of a cell. Essentially, a protein is, at its simplest, a very long chain of individual units, called amino acids, bound to each other by peptide bonds to form an amino acid chain. They sort of resemble a string of beads, and they get twisted and folded into a final protein shape. To make a protein, we need to get to know 2 things: the ingredients, which are the amino acids, and the recipe, or how the finished amino acid chain folds into the protein. Humans use 20 amino acids in our day-to-day protein making. So we have alanine, arginine, asparagine, aspartic acid, cysteine, glutamic acid, glutamine, glycine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, proline, serine, threonine, tryptophan, tyrosine, and valine. Phew, that's 20.
जय गौ माता। जय गोपाल। आप देख रहे हैं गोकुल धाम गौ सेवा महातीर्थ झज्जर हरियाणा में।
One way to divide these is into the ones that we make ourselves and the ones that we cannot make ourselves. There are 5 amino acids that are dispensable: alanine, asparagine, aspartic acid, glutamic acid, and serine, because we can make them de novo ourselves at any time, and in good quantity. Then there's 6 of them that we call conditionally essential, because we can make them most of the time, but not always. And these are arginine, cysteine, glutamine, glycine, proline, and tyrosine. Finally, there are 9 of them that we cannot make ourselves: histine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. And as a result, we have to obtain them from our diet. We call these the essential amino acids. Alright, so the amino acid. Just from the name, you can tell they've got an amine group, or NH2, and also an acid, in this case a carboxylic acid group, or COOH. The amine and carboxylic acid groups are both bound to the same carbon, called the alpha carbon. Now, at a physiologic pH of 7.4, the amine group has a positive electrical charge, and the carboxyl group has a negative charge. Having both a positive and a negative charge makes amino acids a type of Zwitterion, which is German for hybrid or double ion. Now, the alpha carbon also has a side chain, sometimes marked as R. And this side chain gives the amino acids certain properties, which can play an important role in the overall protein structure. First, the side chain can be hydrophilic or hydrophobic, so water-loving or water-hating. Hydrophobic amino acids have non-polar side chains. This might be in the form of an alkyl side group, which is a saturated hydrocarbon, seen in valine, glycine, alanine, leucine, isoleucine, methionine, and proline. Alternatively, it can be in the form of an aromatic side group, which involves a 6-carbon ring, like in phenylalanine, tyrosine, and tryptophan. Now, hydrophilic amino acids have polar side chains. These polar side chains might be acidic, like when their side chains contain additional carboxyl groups, like aspartic acid and glutamic acid. Other hydrophilic amino acids have polar side chains that are basic, like lysine, histidine, and arginine. At physiological pH, the acidic groups lose a hydrogen, and the basic groups gain a hydrogen. Finally, some polar side chains are neutral. For example, they can contain hydroxyl groups like serine, threonine, or tyrosine, or sulfhydryl groups like cysteine, or carboxamide groups like asparagine or glutamine. Now, keep in mind that the charge on an amino acid really depends on its side chain, as well as the pH. For example, at a very low pH, the amine group is positive, while the carboxyl group is neutral. And at a very high pH, the amine group is neutral, and the carboxyl group has a negative charge. And at a pH that's somewhere in between, both groups are electrically charged, and they cancel each other out, resulting in no net charge for the amino acid. The just right pH, also known as the pI, or isoelectric point, is different for every amino acid, and it depends on the specific side chains. For amino acids to link up in a chain, the carboxyl group of one amino acid has to bind to the amine group on another amino acid, creating a single peptide bond. This is a condensation reaction, meaning that 2 amino acids are basically smushed together, and the OH from the carboxyl group along with one of the hydrogens from the amine group get released as a water molecule in the formation of an amide bond. While technically being a single bond, it actually has the properties of a structurally stronger double bond, thanks to the property of resonance. Now, resonance is a property of a molecule where electrons get shared across the molecule, while keeping the arrangement of atoms the same. Basically, the electrons from neighboring functional groups of the amino acid are borrowed, and that makes peptide bonds stronger and more stable. So amino acids are, essentially, a carboxyl group, an amine group, a side chain, and a hydrogen all bound to an alpha carbon. Now, there's an interesting geometric property here called chirality, which means that each amino acid can exist in two forms that look like mirror images of each other. These two forms are called enantiomers of each other. We have the left or levo-oriented amino acids, as well as the right or dextro-oriented amino acids. While similar, these are definitely not the same. Think of it like a pair of shoes. Even though they're made out of the same materials and generally look alike, the left and the right shoe are not interchangeable, at least not without a lot of pain involved. As it turns out, proteins are only made out of levo-oriented amino acids. Now, protein production itself happens in cellular structures called ribosomes, which use the messenger RNA, which is essentially a...
We will discuss the protein synthesis and protein folding maybe in the next session, not in this class. The overall objective of today's class was to understand and know our amino acids, different amino acids, which gonna ultimately make proteins, and these proteins will ultimately be part of our membrane, and the second most abundant component of a membrane, right, up after lipids. So this is how we gonna learn about the membrane proteins. If you guys have any queries or anything, you can always write it to me and I'll try to clear it ASAP. On the other hand, thank you for your time and attention. Thank you. Bye bye.